All right, today we're going to jump in our little time machine and set the dial for 1985. A time when brain science was on the brink of something huge, and a team of researchers was about to draw a map that would literally guide neuroscience for decades. So let's dive in and see how they pulled it off.
So, we've all heard of serotonin, right? It always gets called the happy chemical. But what if I told you that's, well, a massive oversimplification? The real story is so much more complex, and it all boils down to where in the brain this chemical is actually doing its job. Here's a great way to picture it. Think of serotonin as a master key. But the brain isn't just one big door, right? It's like this enormous mansion with millions of different rooms, and each room has a very specific, unique lock. Scientists call these locks receptors. Now, the key only works if it fits the lock, and which lock it opens changes everything. The big question back in 1985 was, where in the world are all these different locks? To figure that out, we're going to follow the exact trail these scientists left for us. First, we'll look at the puzzle they were trying to solve. Then we'll check out the super clever tools they used to, see something that's totally invisible. We'll uncover their story of two very different receptors, see how they checked their work by comparing humans, pigs, and rats, and finally, we'll get into why this brain map from so long ago is still a huge deal today.
Okay, so let's set the scene. Imagine you're an old-school explorer, but the map you're using just has these giant, blank areas labeled, "Here be dragons." That is exactly the situation scientists were in.
So they knew that these two specific serotonin locks, or receptors, their technical names are 5-HT1C and 5-HT2, they knew they existed. They just had absolutely no clue where they were in the human brain. And if you don't know the location, it's impossible to even start guessing what they do. Are they in the part of the brain for memory or emotion, or something as basic as keeping you alive? It was a complete and total mystery.
Now, to fill in those blank spots on the map, you can't just, use a telescope. You need a really specialized toolkit for exploring inner space.
And the first tool they used is it's brilliant. Scientists created these special keys called radioligands, and these are molecules that are engineered to fit perfectly into only one type of receptor lock and nothing else. But here's the really clever part. They attached a tiny radioactive tag to them, which basically made them glow in a way their instruments could see. So then they used a process that's pretty much like photography, but for molecules. They took incredibly thin slices of donated human brain tissue, they bathed those slices in these glowing radioactive keys, and then they laid them right on top of photographic film. So wherever the keys found their matching locks, they stuck, creating a little radioactive hotspot that exposed the film. And when they developed that film, bam, they had it. A direct picture showing exactly where these receptors were hiding in the brain.
So after all that incredible work, the invisible finally became visible. The map started to fill in, and boy, did it tell a fascinating story.
I mean, just look at this. The results were immediately striking. On the left, you've got the map for the 5-HT1C receptors, and on the right, for 5-HT2. They're in completely different neighborhoods of the brain. It's like finding one inside a power plant and the other inside a university library.
First up, let's talk about the 5-HT1C receptors. They were just packed, super densely, into this area called the choroid plexus. And this is the part of the brain that produces the fluid that cushions and cleans our brain and spinal cord. This was a massive clue that maybe serotonin helps manage the brain's most fundamental environment.
Meanwhile, the 5-H2 receptor, it showed up somewhere totally different. The frontal cortex. That's our command center. It's the part of the brain responsible for our personality, for reasoning, for complex thought. Basically, the stuff that makes us, us. This discovery was huge because it was the first to directly link this specific receptor to our highest cognitive functions. And in their paper, the scientists could finally just state it plainly. I mean, this quote might sound a little dry, but this is the scientific equivalent of planting a flag on a newly discovered continent. They had set out to find these receptors, and they did. They'd officially put 5-HT1C and 5-HT2 on the map of the human brain. But, a good explorer always double-checks their compass. After mapping the human brain, the very next step was to see how it stacked up against other animals. And this is an absolutely crucial step for any future research.
Yeah, this is a critical question in medical science. If you want to develop and test new medicines, you often need an animal model where the brain works a lot like our own. So the team had to ask, is the map we just drew for humans the same in common lab animals, like, say, pigs and rats?
And this chart, wow, it just lays out the answer for the 5-HT2 receptor perfectly. It's comparing the human and pig brains, and every single one of those little dots is a different chemical. See how they form an almost perfect straight line? Well, that means the lock in the pig brain and the lock in the human brain are practically identical.
But the story was different for the rat brain. The results showed that the pig brain was a really strong match, which made it an excellent model for future research. The rat brain, on the other hand, had some pretty big differences, making it a less reliable guide for this specific system. And for scientists trying to develop new medicines, that was a hugely important practical finding.
Okay, so we have a map, and we've confirmed it's reliable. But what's the ultimate legacy of this 1985 expedition into the brain? I mean, why are we still talking about it all these years later?
Well, the impact was just enormous. This study gave us the very first detailed map of these two key serotonin receptors, and by showing they live in different parts of the brain, it helped prove they have very different jobs. It gave researchers a much better animal model to work with, and, maybe most importantly, it paved the way for designing smarter drugs, medicines that could target one type of receptor without messing with the other, which could lead to way more effective treatments with fewer side effects. For over 35 years, this foundational work has been a cornerstone of neuroscience. It is just a perfect example of how basic, curiosity-driven science, just trying to fill in a blank spot on a map, can completely change the course of medicine. Before this paper, our understanding was, well, it was fuzzy. It was incomplete. And afterward, we had this clear, detailed landscape. It was a guidebook that allowed countless other scientists to launch their own explorations, leading to so many of the treatments we have today for conditions like depression and anxiety.
And that kind of leaves us with one final thought. This incredible map was drawn almost 4 decades ago, using what was, at the time, totally state-of-the-art technology. It really makes you wonder, with all the amazing tools we have now, what uncharted territories are left? What blank spots on the map of the human mind are today's explorers working to fill in right now?